I'm Berman. If you've ever sat through a bio-team talk, you know that we all have a disease of trying to cram a whole lot of stuff into a small amount of time. And so my speaking rate's going to go about double or triple here in a second because I only have 52 slides for about 22 minutes, so this should be interesting. Today I'm really going to talk to you about sort of the requirements of HPC in life sciences because it's really a non-trivial thing to do. Currently I'm the general manager of government services at BioTeam. I also do quite a bit of consulting there. As Jack said, I'm basically a fallen scientist. I spent 12 years in the laboratory doing molecular biology and neuroscience work, 2 postdocs, one in bioinformatics. But I've also been developing HPC and infrastructure for 17 years. And so the whole goal for me has been to enable science. BioTeam, we're a very small independent consulting shop. We're basically staffed by scientists who are forced to learn IT, software, and HPC to get our own research done. We consult in infrastructure, informatics, software development, cross-disciplinary assessments, and the company's been around for about 13 years. We basically bridge that gap between IT, science, and high-performance computing, and we are in and out of a lot of places. And really what we've been focusing on lately is this whole idea of taking information from a laboratory and helping scientists convert it to knowledge. And these days, with the computing needs of all of this, that involve network, it involves compute, it involves storage, it involves software and analytics, and they're all plugged in along this pipeline somewhere, right? The problem is that it becomes really very difficult for any scientist to navigate that. Sorry. Get back to where I was. Okay. It becomes very difficult for any scientist to really navigate that because all this infrastructure is very complex and they may or may not have the support to do it. And so what we try to do is sort of bind it all together into what we're calling a converged solution. Not converged in the normal sense that everyone talks about where, two things are fused into the same architecture. We're talking about this is basically a tool that laboratories use to get to an endpoint, and hopefully in the end we've got sort of happy scientists, right? So with that in mind, why am I here talking to you today? The reality is that we go in and out of about 45 or so different organizations a year. And so we end up getting this really unique perspective across much of life sciences. Since we're really an unbiased third party, we really see just about everything that you can imagine. So everyone's heard it. Big data has arrived in life sciences. Data is being generated at crazy rates in life sciences. Research and biomedical works have been completely caught off guard because, the way they see it, last year they were on Excel. Right, using USB drives to get all their work done. They can't do it anymore, and so IT basically is trying to run to catch up. But the organizations don't want to invest in this infrastructure necessarily. So money is tight. Organizations don't want to invest. The reality is that about 25% of all bench life scientists are probably going to need HPC in some way in 2015. Right, that's an enormous number of scientists. Very few of whom have ever used a command line ever. Right. And so on top of that, research is really, really hard, anyone who's done it, right? And in general, because of this new sort of data push, it's being made harder by a lack of support and a lack of sufficient infrastructure that actually works for scientists. And so they get frustrated. They do stupid crap like this, right? Where they go out to Best Buy and buy a whole bunch of drives and pile them up next to the sink in the lab. Right? All their data is on this, right? This happens. And they will build a cluster under their desk if it helps them get their job done. It's going to be slow, it's not going to be good, but that's what they're going to do. In general, because the mission of all these organizations is the science they're trying to drive, they eventually will win against IT. It's just going to be a painful battle. So as such, it turns out it's a pretty risky time to be doing HPC for life sciences. The big picture of the problem is that there's been this huge enormous growth in the rate at which lab equipment has been growing, right? Way, way over cubic. Platforms are being redesigned, improved, and they're generating more and more data, and scientists love more data, so they keep collecting it. The problem is that IT is not part of that conversation in general, right? Scientists go out with their budget and they buy a piece of equipment, it generates terabytes of data a week, and then IT finds out about it. right? And so there's no conversation there. And so the reality is that beyond that science, at least in life sciences, is progressing at a rate way faster than IT can refresh or change. The instruments and protocols are refreshing on the order of months, where IT's general life cycle refresh is on the order of years, 2 to 7 years. And so the trick here is that we have to design systems today that support sort of this really unknown research environment that's coming up sometime in the next 3 to 5 years. That's really scary because there's no real use case for that. How do you do that, right? And the reality is, what used to work well as a couple of Macs in a lab now looks like this. Right? This is essentially the new normal for informatics and life sciences. This is new for life sciences. Those of you who have been in other, high energy physics or anything else, this has been normal for a long time. But for life sciences, we just got here. The reality is that if IT gets this wrong, it results in lost opportunity, missing capability, frustrated and very vocal scientific staff, and problems in recruiting, retention, publication, product development. And so what's really driving this today? I think everyone's really heard about genomics, right? This sort of revolution in the way that we can read genomic sequences and analyze them. Next generation sequencing, or NGS, is what it is. So for those of you who aren't biologists, what's the big deal about DNA? It's kind of like the hard drive of life, if you really think about it, right? It's a template of life. DNA is read, it's turned into RNA in a cell. RNA is read, and it turns into proteins. Okay, proteins are basically, they're the functional machinery in a cell and are basically allow you to do everything you do. So the idea is that understanding that template might help you understand the basis for disease, although there's a lot of steps in between. So I'm not going to go through this in real detail. This is to illustrate how one way of doing next generation sequencing works, to illustrate where the data is coming from in a sense, right? So you basically have genomic DNA. It's broken up into lots of little pieces. It's put onto a slide, essentially, and then in a careful series of reactions, fluorescent dyes are bound to each of these little pieces of DNA in order, and then an image like this up in the corner is taken at every stage in the reaction. The colors are red, they're isolated, and those little colors correspond to a nucleotide or a base, and you end up with little bits of sequences. And so next what you have to do is you take all those little bits of sequences and you sort of figure out how they fit together, and then you collapse it down into this consensus sequence, and that's sort of your genome or your sequence, right? That's hard, and turns out to be a very non-trivial compute exercise. Then you run it through some algorithms to figure out where the disease is, and see how it matches to other things. And this whole section that I'm glossing over here is really where all the HPC is used in life sciences right now, at least with genomics. And so when you think about the scale of this, the human genome is basically, it's a gateway to personalized medicine, as they say. The human genome has 3.2 gigabase pairs, okay? 23 chromosomes, about 21,000 genes, and there are over 55 million known disease variants in the human genome, right? So it's actually a pretty large problem, and this changes from person to person individually quite a bit. So the real problem, and why next generation sequencing is a primary driver, sequencers are actually very cheap now and very fast. So never in the history of life sciences has it been easier or less expensive to generate enormous amounts of data than it is right now. Some of these sequencers can generate an entire human genome in 18 hours for $2,000. Right? By comparison, the first genome took 13 years and $2.7 billion to create. This was 12 years ago. Okay, so everyone's doing it. The raw data that comes off these things can be 3 terabytes in size in that amount of time. And here's something really scary. The picture here, the MinION, that's a sequencer. It's this big, all right? Doesn't particularly work that well yet, but it's getting there. And that thing generates enormous amounts of data. It plugs into the USB port on your computer, right? So this is getting miniaturized, it's getting commoditized, and it's happening at a scale that no one's ready for. The reality is that other methodologies aren't far behind. There's high throughput imaging, robots screening millions of compounds all the time. There's deep layers of imaging. These things are in the tens of terabytes per week. High resolution medical imaging, looking at 3D volumes of structures and comparing them, also tens of terabytes a week, and it's a really huge problem. It's causing this real deluge in data in the tens of petabytes. NIH at the moment is probably generating about 1.5 petabytes of data per month at NIH. That's not what they're funding, that's just locally at NIH. And so, they have this real need for really fast networking. These are pictures of what 100 gigabit ports might look like, but they don't have enough storage or compute, and nobody does. And then there's more, right? Data and file types. Anyone who's dealt with storage or high-performance computing, this is a real problem, especially in life sciences. Life sciences has everything. We have massive text files. We have massive binary files. We have flat file databases, spreadsheets everywhere, directories with 6 million, 17 million files I've even seen, large files that are 600 gigabytes plus. I've actually seen one that was 4 terabytes, believe it or not. And lots of small files that are 4 KB or smaller. And so that's a real challenge to deal with all those data types and any sort of hardware that you throw into a system like this. And then on top of it, the application characteristics are all over the place pretty equally, right? Most of life sciences, a lot of it is really SMP threaded applications bound by I/O or memory. Okay, there's hundreds of apps and codes and toolkits. There's some things that run on that need 1 or 2 terabytes of memory, but run on a single CPU. Okay, so it's all memory, it's all I/O. The vast majority of stuff in life sciences is Perl, Python, and R. MPI is really, really rare, and well-written MPI is even rarer. Okay, I think that's true in most things, but in this case, it's a little on the ridiculous side. And there's a few MPI apps that actually benefit from expensive low latency interconnects, like chemistry and molecular modeling and things like that, that the organizations don't tend to invest in, right? And so, with that background, what do you need to do to be able to create HPC in life sciences? The first thing, and I think this is true of just about everything, and it's a hard pill, I think, for this particular community to swallow, is build it for the use case. You can't just build a machine for performance, right? It's fun to get on the top 500. It's awesome, it's shiny, but the reality is that doing that is short-sighted, especially in life sciences. The systems that are being built really need to solve the problems, not create more problems for the scientists. A lot of times when something's developed for performance, it's like a Porsche, right, or something, right? You drive it for a while and it breaks all the time, right? And it makes it really hard to figure out how to use. And so this means that first and foremost, before you start engineering something, you have to understand the use case before designing it, right? The reality, though, is that if you actually build something that solves their use case, at least in our experience, you have a lot more fun in the long term. And so, the use case of life sciences, there's no other way to put this other than life scientists are basically the thugs of HPC. If you have it, they'll break it, and they'll break it hard. As I said, they have every kind of file you can imagine. They have every kind of algorithm, good, bad, ugly, and worse. They want it all right now. They keep everything. They're total hoarders. Some of it's by mandate. They certainly don't want to pay for it. And the reality is, though, your health may eventually depend on it. Right? We have to solve these problems for humanity, and compute is the way to do it. So what do we see in the field right now? The reality is that the core compute, it's relatively basic. Really, Linux compute clusters, the baseline platform with commodity servers. Not a ton of capability supercomputing in this case. The interesting thing is that capability supercomputing is actually a growing need. Life sciences is starting to catch up because we have these petabytes of data that are sitting there doing nothing because it has nowhere to go. And some of our lab instruments actually know how to submit HPC jobs to schedulers now. So it's just, pipelines just go off, right? And so the weird thing about any new compute coming out here now is that people are really starting to pay attention to the power draw. As chips become slightly more power efficient, but you need a lot more density, power efficiency is really a requirement as you're in the half to 1 petaflop plus range, 30,000 plus cores. And so what's really happening when people are designing these things is that high core density in multi-server racks is really key. You get better distribution of power. Custom chassis with extreme densities are the best at power efficiency per core. And then on top of that, with compute, at least in life sciences, you need a really high diversity in the type of nodes that you use. So new and refreshed systems, you end up with these really heterogeneous type of clusters, right? Homogeneous systems just really don't work in life sciences because of the laundry list of use cases that I listed earlier. So the newer clusters are sort of a mix and match of things. We have fat nodes with tons of CPUs. We have thin nodes that have really fast CPUs, large memory nodes, 1 to 3 terabytes, GPU nodes for compute and visualization, co-processor nodes like Xeon Phi and FPGA, and then you have analytics nodes with like SSDs and Fusion I/O and Flash or large local disk for big data tasks and things like that. And then, when you're really thinking about this, these are happening in pretty extreme density. These are 8 NVIDIA GPUs shoved into one machine, right? And that's a reality now. The power draw and the amount of throughput you get through that is pretty amazing. And the high memory machines, 96 DIMMs shoved into a single box, everywhere you can imagine, right? But that's how you get to 3 terabytes in a single box, and it matters for most of life sciences. So none of this stuff is actually particularly hard. It's all doable right now. The really, really hard thing right now is storage and data management, all right? It's the biggest headache for sure in life sciences. Requires connectivity to HPC labs and externally for data sharing. It needs to be scalable. Life scientists, are really well into petascale data, as I said, but it's every sort of file type you can imagine, everything from small to large. And so they really are starting to need performance. Life scientists have been fine with running off of their USB drive for ages until they have many, many petabytes of data. And so large HPC really needs this. And as I was talking about before, life sciences code is really very I/O bound. It does a huge number of read-write calls, a whole lot of random I/O, even though CPU utilization may be as low as maybe 20% on a single core, right? The actual load average on the machine will be through the roof because of all the data I/O going through it. And if you don't architect your systems right and you have any sort of a scalable system, this code, just a couple of them, will bring your storage to its knees in no time. And then no one will be able to use the system, right? So you have to really architect for this. IOPS are critical in 2015 for life sciences because most of that code is really poorly written. The problem is you're not going to get anyone to redevelop it, so you have to build for that use case, right? And so ideally, given how bad things are in how people are treating their I/O, you really need a throughput per core of about 100 megabytes per second. That would be ideal. And if you're dealing with just to illustrate how unrealistic that is, if you have 10,000 cores in a system, that's a terabyte per second transfer overall, okay? It's just not realistic. You can't get that. You're lucky to get 10 megabytes per second per core on any sizable system. So the type of storage we see out there these days really are NAS, scale out NAS. It's the easiest option for most people. That's where everyone's home directories are. That's where they store their data, and a lot of HPC runs off of this. The problem is it doesn't really deliver performance to core, and so since we're sort of edging into this space of really needing real compute, parallel and distributed storage is becoming much more standard in life sciences. In life sciences, GPFS holds the major portion of the market that we see. Most people use that. Lustre is really gaining ground, but it's worse than GPFS with small files, and small files are a real problem in life sciences. But GPFS also isn't great with small files, depending on how you architect it. And so global buffers, IOP tiers that sit in front of these storage are starting to gain some ground, but it's really a new thing where these arrays of solid-state disks basically sit in front of whatever file system there is, take all the random I/O, buffer it, and re-output it in an ordered I/O, which keeps the system running. Dedicated IOP tiers that have millions of IOPS per second at scratch. There's a whole lot of object stores coming out there, sort of almost a tier 3 type of storage, an archive tier instead of backup. Things like erasure coding and pods all over the place are making it so that, okay, if you're done with it, put it here. Otherwise, it's not getting backed up. And then there's still a lot of local SSDs for scratch. So the other thing to really think about here is that if you're going to build a system like this, the cluster networking really starts to matter, right? If you need throughput from storage, then you need a back-end network that's going to do it. In life sciences, up until, 6 months ago, everyone really thought that 1 gig and 10 gig Ethernet networks worked just fine for life sciences. And the reality is that it just isn't going to cut it anymore. So building I/O, as I said, requires a very high-performance network. Most of the things we see are from InfiniBand at the moment. FDR and EDR, and there's a lot of advantages to basically eliminating traditional Ethernet on these systems. It's a high-speed network. Overall, it's a much lower cost than Ethernet. FDR is 56 gig plus. You can use IP over IB to sort of bind these two fabrics together. You lose some performance, but with life sciences, that's okay. And the whole cluster can basically work for all job types rather than having a certain section that's InfiniBand and a certain section that's Ethernet, or having to manage two networks on the same backplane. And then when you're building these things, the cluster topology matters a lot. And so what we see a lot in anything really over 5,000 cores is this idea of a fat tree network on the back end that is very scalable and goes from, basically the pipe increases as you go up so that every lower end can be saturated. And the nice thing about this is that if you build it this way, then you can build it so that you can expand in units pretty easily without doing any recabling of the back end network. And so for cost savings, most of these things end up being 2-to-1 blocking, but you can build them 1-to-1 blocking with some interesting combination of technologies. You can build in gateways that talk outside of this, so you don't need Ethernet at all, right? Mellanox actually really enables that with some VPI and gateway stuff that they have on their switches. You can hang data transfer nodes off of these and use them as fast data transfers and storage at the root. So why go to all this trouble? 10 gig is fine. That's true for now, especially in life sciences, but faster than 10 gig doesn't really help most bioinformatics code, but at scale things get really messy. And so the landscape is changing, code is being optimized, data sets are still increasing in size, and the lab tech is still developing off the scale, and so you have to build for the unpredictable. I'm running low on time, so let me skip through this a little bit. We really see a lot of, as far as the software stacks out there, we really see a lot of SGE, Univa sort of taken hold. Slurm is out there, Torque and Maui, and we even still see some PBS Pro and LSF, but really SGE is the biggest proponent of schedulers out there. What's really interesting is that the key features that are really looked for are resource mapping, so GPUs to CPUs, being able to schedule individual GPUs that share a lot of them in a box. Core-based scheduling, rich resource management, threads, memory accelerators, mixed environments. Meta-scheduling and hybrid environments is really important. Application-aware scheduling is becoming big. And things that are not out there, really. Hadoop has been really slow to adopt. There's lots of talk and very little walk, not a lot of dedicated instances. It tends to live on core storage when it's out there to make it schedulable so you don't have to have dedicated infrastructure for it. But the next stage then is getting data to and from a cluster or the storage on the cluster. And so you really have to build 40 gig and 100 gig infrastructures to get data in and out of there from any sizable organization. And to do that, what we've seen a lot of is the development of this concept of a science DMZ, which is a very fast, low friction network that sits outside of an enterprise network. The reason you do this is because, as most of you know, enterprise networks aren't going to lower their security level to allow you to run data through it. And most of life sciences data isn't regulated or have any personal health information in it. And so that's the central premise there. And so really, what's the future look like for HPC? It's designing for the use case, first of all, but also, given tight funding environments, a lot of locations are really starting to talk about hybrid HPC, right? Hybrid cloud, right? It's kind of a new idea where you have sort of a small local footprint and then a large dynamic, scalable, and orchestrated cloud environment in a virtual scale somewhere else. You use meta-scheduling and DevOps in between to sort of synchronize them, and there needs to be a high-speed network between them. But overall, this could be way lower cost, especially in lifecycle management moving forward, as long as it's automated enough. So there's not a lot of working examples yet, but it is definitely coming. So the thing is that all this stuff actually has to work well together, right? And this is what I started with. And so unless all these things string together well into a unified architecture, it really doesn't mean anything. It's really hard to use. And so creating sort of an end-to-end solution for the use case, which in this case is science, equals a converged infrastructure. And so this is what I started with, was this whole thing, right? And so if you end up with a converged solution so that works well and helps the laboratories actually take their information and knowledge, then you have happy scientists and happy people. The thing that we don't really talk about when we're talking about all this is that people matter too. You have to shove people in there. They have to be a part of that converged solution, both from a support and analytics, people who know how to install and optimize scientific software, that sort of thing. And then the next thing to really think about, though, is that there's a new bottleneck when you actually fix this, and that's the laboratory. Right? Data coming out of the laboratory. Laboratories are not architected for data to pull out and go anywhere useful. In fact, it's very rare that you have reasonable Ethernet ports in in laboratories. And so maybe the not so distant future is to actually organize all this infrastructure around the laboratories and the data that's coming out of it. High energy physics has this figured out because their experiments are so gigantic that you have to build for it, right? The life sciences isn't far behind. Then hopefully what we end up with is just learning knowledge, and we get happier and better. All right, not too bad. That's it. Thank you.